So now let's go over how secondary growth occurs in woody species of plants. Keep in mind that secondary growth occurs in most gymnosperms, such as pine trees, and many eudicots. But it doesn't occur in most monocots. Secondary growth is also restricted to particular portions of a plant. It occurs in stems and roots of woody plants, but very rarely in the leaves. The tissue that's produced through secondary growth arises from the two lateral meristems, the vascular cambium and the cork cambium. Keep in mind that primary and secondary growth can occur at the same time in the same individual plant, but in different locations. To illustrate this point, let's look at growth in a young stem. The base of the stem represents the oldest portion. The region closest to the tip of the stem is the youngest, less than 1 year old. So after cellular differentiation begins, just behind the apical meristem, which isn't pictured here, we get a few layers forming in this stem. Okay, we have the pith, and the first vascular layers that are put down are the primary xylem and the primary phloem. These are the tissues that form during the first year that this stem is present. And they originate from this region, the vascular cambium, denoted here in red. Because this is a young stem, we have an epidermis. Okay, remember that's a single layer of cells. Eventually, that epidermis will mature, and other layers of cells that will be added to it will cause it to toughen up and change. So this young material is extending further and further in this direction. Okay, that's primary growth. Last year's growth and the year before that are exhibiting secondary growth. This region of the stem is no longer growing longer. Only this green portion here is extending in length. So if we were to put a nail in this portion of the stem and come back years later, it would still be in the same place due to the lack of elongation in this region. So let's look at how the width of this stem has changed in the past year. Okay, we have additional tissue being added from the vascular cambium. The primary xylem and the primary phloem has not changed, but we see the addition of the secondary xylem in a ring here and the secondary phloem. So this new tissue is being added at the point of this red line, and as we move towards the center of the tree, the xylem gets older and older, but the phloem is formed in the opposite direction. The older phloem is found towards the outside, and the newer is formed towards the inside. Okay, keep in mind that the newest of both types of tissues is the closest to this red line. And in the 2-year-old stem, we see a little bit of a ridge here as a second ring of xylem has been added. And the distinction between the 2 years of phloem is not as obvious. although it's definitely true that this layer has gotten a lot thicker. So the portion of the tree that we call wood, from this point on to the center of the tree, is composed of xylem. And everything on the other side of the vascular cambium, including the primary and the secondary phloem, as well as this periderm layer that's also changed since the first year of growth, is called the bark. So notice that this periderm is enlarging from the epidermis as additional cell layers are put down. So we could say that as a tree grows, both the wood and the bark gets thicker over time. So keep in mind that the vascular cambium is a thin ring of meristematic cells. Okay, meristematic means that they're able to divide, and that the cells found in that ring are currently undifferentiated. The cells found in this ring are also called initials. And they're depicted here by the letter C. Okay, every time one of these C cells divides, one of them remains a C with the ability to continuously divide and differentiate, and another one is going to become an X or a P. So this vascular cambium adds the X or xylem cells to the inside. At the same time, it's adding the phloem cells to the outside. The layers of xylem are much thicker than the layers of phloem that are added each year. And it's the xylem that's responsible for these distinguishable growth rings that can be counted to age a tree. So let's take a closer, even a microscopic look at what these annual rings look like in an actual section of a tree. Okay, the vascular cambium is this thin line shown here. The oldest growth rings are down towards the center of the tree, and the newest growth rings are the closest to the vascular cambium. All different types of water-conducting cells from xylem tissue are present in each of these annual growth rings. The cells that were put down early in the season, usually during the spring, are found here. Okay, these cells are going to be the largest. They have the most cytoplasm and the thinnest cell walls. Later in the year, in the summer and the fall, the new cells that are put down are a lot smaller and have a thicker cell wall. And it's these differences in the shape and the constitution of these cells that's responsible for our ability to tell the annual rings apart. Generally, in temperate areas, the tree isn't growing over the winter. Now, it may not be possible to distinguish between annual rings in tropical trees. And this is because there may be no difference between the late wood and the early wood if a tree is found in a region with very similar growing conditions year-round. Also notice these lines running perpendicular to the vascular cambium. These are called vascular rays. And these are narrow bands of parenchyma cells that facilitate connections and the transfer of materials between xylem and phloem. So it's not only the vascular cambium that's responsible for secondary growth, but this additional meristem, known as the cork cambium, indicated here by this thin dark blue line, also increases lateral growth outward. Remember that when a young shoot forms, only a single layer of an epidermis is present. Starting in the second year of a tree's growth, these cork cambium regions result in the deposition of additional tough cells that contain a waxy substance called suberin. After these cells die, they start to build up and produce a substance called cork. This establishes a new layer that replaces the epidermis, called the periderm. It's a very tough, thick, protective layer, and it's impermeable to things like water and gases. Now, both the secondary phloem and the periderm make up the familiar structure that we know as bark. Although bark is a very helpful substance in protecting a tree, even from severe natural disasters like fires, one of the problems is that the trunk of a tree does need some gas exchange. So an adaptation to overcome this problem are these ridges in the bark, known as lenticels. Lots of woody species have them, but they're especially obvious on species like this cherry tree, as well as some other related species like plums. So the practice of aging trees based on their annual rings is known as dendrochronology. And while I don't expect you to memorize this term, it's a very interesting science. And it's one of those interesting ways that biology can be used across different disciplines. Dendrochronology is not only of interest to biologists, but also climate scientists and historians as well. Let's use this example of an extremely old Douglas fir tree that was found in Arizona. After counting the years back from the year in which this tree was cut down, scientists were able to label the year 550 AD. Keep in mind that many trees, particularly gymnosperms, often live for hundreds or maybe even thousands of years. The growth rings in general, from the center to the outside, are getting smaller. Okay, that's typical because plants don't grow quite as quickly when they're older as they do when they're young. But we can tell a lot about what was going on in particular years by comparing the width of rings to those around them. So we notice that this ring right here, as well as this one, and this one over here, are a lot narrower than the rings found on either side of them. So as you could guess, that was probably a very dry year. Compare with years earlier or later than it. In this way, biologists and climate scientists can work together to study weather patterns over hundreds of years. It's also really fun to go into a history museum and see the rings of the tree labeled with various significant historical events from far in the past.